So the session we have in this afternoon about circulating biomarkers with a twist, focusing on circulating DNA and circulating tumor cells, is going to bring together some speakers from around the world and the UK to look at what's new and upcoming in these fields, which are subject of very hot research and very fast-moving fields these days. Well, it's the coming together of technologies and the applications, and these are fields of research which rely heavily on genomic analysis, and the methods of genomic analysis have now made it possible to really study with the kind of resolution that allows us to pick up mutations coming from cancer, even in complex fluids such as plasma DNA that contains a lot of background DNA in addition to DNA coming from cancer, as well as the methods that are required to look at single circulating tumor cells, which require kind of sophisticated technology and are now really developing to the point where these are becoming much more useful and practical. In my talk, I'll present an overview of progress and a review of where we see things lie, where the field has developed to, and where we think it's developing to. We won't be presenting any groundbreaking culmination of big studies in today's talk, rather an accumulation of incremental steps that are slowly progressing us towards better understanding of this phenomenon and what we can do with this for cancer analysis and cancer applications. We're looking at how we can use circulating tumor DNA to monitor cancer response to therapy, looking at circulating tumor DNA as a quantitative measure of tumor burden and how we see that changing over the course of treatment in response to therapy as the patients are initially responding. And unfortunately, if the patients relapse, we can see that reflected in the increasing levels of circulating tumor DNA. I'll be reviewing some of the data that will be presented, and some of it has been published by the other speakers of today, looking at how circulating tumor DNA could be a marker for the risk of relapse after potentially curative surgery or treatment. We'll be looking at how circulating tumor DNA could be used to reflect and to analyze the heterogeneity of cancer where you have multiclonal metastatic disease. In addition, I'll be looking at potential new ways where we can develop circulating tumor DNA further, how we can increase the sensitivity for analysis of low-volume disease, for example, by pooling together multiple patient-specific mutations as a more sensitive way to analyze the amounts of circulating tumor DNA in the blood. Thinking about how this can lead on to future applications in earlier cancer stages for minimal residual disease, and potentially in the future to help in earlier diagnosis of cancer in cases where there are suspected symptoms, and eventually in the future possibly towards kind of screening applications. The analysis is based on material we collect from a single blood test, although at the moment many of these applications are still in research phase, so we try to get the best information we can by triangulating the data we get from the blood test with data that's obtained from tumor sampling, using that to learn both about what we can do with these circulating tumor DNA, helping us to develop the methods further. And the idea is that as we become more proficient with these, then we can make better collect better information on the cancer without the use of the tumor DNA, both as a research tool to look at new resistance mechanisms, for example, to therapies, and also as a diagnostic tool to help identify what's happening to the patients and what might be the optimal care for them down the line. I think it can go in many different ways. I think it's a piece of information that the oncologists can use, and again, we are working together with oncologists and with the new technology that we can bring to see what is the optimal way to use that. I think one possible way to use that is to see if patients are progressing and to change therapy earlier. That kind of change in standard of care would require quite a significant level of evidence if you want to change while the patient doesn't have increasing symptoms, for example. An easier place to apply this is when you already know that you want to make a change in therapy, and you can use circulating tumor DNA or circulating tumor cells in the future to inform about what might be the best next course of action by getting a picture of what the tumor looks like from the DNA that's in circulation. Now, oftentimes you'd like to get a repeat biopsy of the cancer if you are able to, because cancer evolves and changes over therapy. But if you're unable to obtain that due to complications, cost, logistics, then you might be able to get information from the blood that will still be of informative value. It's not yet entered into clinical practice. It's now starting to enter into kind of select clinical trials, which would use this initially as a comparator. Eventually, I expect and hope that in the near future, this will already start to be used to guide some of the upcoming clinical trials, where this kind of information can be used to select treatment within the context of a clinical trial, and these clinical trials will help us see how much this information is really helpful to guide these treatments. The conclusion of my talk would be really that the technology is now here. We know enough about cancer to know that we do want to get serial sampling of the cancer over time. Technology now allows us to do that, and this now opens up the possibility to do these kinds of clinical trials, which the community is just kind of now starting to put together. I mean, these are significant investments requiring, again, putting together the possible therapies that you need to use as a next step, putting together all the infrastructure that you need in order to obtain the data and the samples, in patient consents, all in time in order to undertake this exercise.